Assalamualaikum to Dr. Balkis. So today we are from Group B and we will be presenting the presentation of practical in downstream processing IBA 405. So first of all, let me introduce our group members. Group B consists of me, SALIHAH binti JEMAIN, AMIRAH binti AHMAD, Mohammed Zharif Asyrani bin Mohammed ALIAS, and JOYCE JEYA BHARATHY anak perempuan BALA CHANTHIRAN. So for our group, we choose research article entitled of A Mechanistic Examination of Salting Out in Protein Polymer Membrane Interaction. First of all, in the article, it mentioned that protein separation and purification are the most common areas in biological drug development, owing to their empirical optimization. Mechanistic insight into protein separation will enable microscale separation to be predicted and would have a wide influence on common healthcare items and also biosensing device. A common process in protein separation is salting out, in which high salt concentration are introduced to the mobile phase to improve elution efficiency. The principle of salting out, in which high concentration of salt will decrease the solubility and the protein will precipitate. This can be explained by the excess of ions where not bound to the protein will compete with the proteins for the solvent. The decrease in solvation allows the protein to aggregate and precipitate. The protein molecules tend to associate with each other because protein-protein interaction become energetically more favorable than protein-solvent interaction. By salting out process also, the unwanted protein from a protein solution combination, unwanted proteins can be eliminated. After removing the precipitate via filtration or centrifugation, the desired protein can be precipitated by altering the salt concentration to the point where it becomes insoluble. That's all from me. I will pass to the next presenter.
Assalamualaikum warahmatullahi wabarakatuh. My name is Mohammed ALIAS, and I'll be taking over from here, where we will talk about the background of the research and also problem statement of the research. For this journal, the background of the research is that the protein separation and purification are the dominant expenses in a biological drug development, largely because they are optimized empirically. And also, salting out is a common process in a protein separation when it occurs when a high salt concentration is introduced to the mobile phase to improve elution efficiency. And by utilizing this salting out process, it will help to separate to help the separation by manipulating the protein stationary phase interaction by ionic shielding. Some of the methods used to monitor protein surface interaction, which includes surface plasmon, resonance, isothermal titration, calorimetry, and atomic force microscopy. There are some problems from the experimental techniques, such as there will be a lack of high spatiotemporal resolution, which is necessary to develop a physiochemical mechanism of the protein stationary phase interactions. A way to overcome this by using single molecule microscopy is optimum to directly visualize the protein mass transport at a wide array of complex interfaces, one molecule at a one time with a bigger spatiotemporal resolution. Now, the total internal reflection fluorescence, or TRIF, is a wide field single molecule tracking which was utilized in combination with ensemble fast protein liquid chromatography, or FPLC, which can relate to the tracking observables of ensemble elution profiles. Circular dichronism, or CD, spectroscopy is also utilized to quantify the structural changes of protein at the stationary phase support, and it relates to TRIF and FPLC result. The RIF microscopy has been applied in many other systems, which includes nanoparticle catalysis, DNA hybridization, kinetics, and also protein transports in a live cell. Membrane-based separations such as nylon have been gathering interest because of the reduced mass transfer resistance, increased surface area for adsorption, and also its lower column costs in comparison to the traditional bead-packed columns. Nylon is also commonly used due to its property of chemically robust, optically transparent polymer which is stable under laser illumination, and it's already utilized in many consumer products. The surface transport modes and also kinetics of transference are quantified at the single molecule level, and it's used to explain the reduction of peak broadening observed in ensemble separation. For the objective of this research is to investigate the peak narrowing in a model transferring nylon system under salting out condition using a combination of single molecule tracking and ensemble separation. For the problem statement, these proposed models extracted from these techniques are likely to oversimplify the underlying mechanism of interfacial adsorption, desorption, surface diffusion, and also surface-induced protein unfolding effects. That is all from me. I'll pass along to my next team member. Thank you.
Assalamualaikum, I am AMIRAH, and next I will explain our results and discussion. First, transferring surface dynamics. Single molecule tracking resolves two distinct modes of transferring surface at nylon, which are tuned by salt concentration. One population exhibits a continuous time random walk CTRW, while the other one undergoes single site adsorption-desorption. CTRWs present in transferring nylon interactions can be identified by waiting time distributions that fit a power law. The prevalence of transferring molecules undergoing CTRWs decrease as salt concentration increase, resulting in more single-site adsorption-desorption. However, CTRWs remain the dominant mode of transferring surface transport. Representative single-molecule trajectories at varied ionic conditions illustrate the two modes of dynamics that transferring displays at nylon. The dynamics are spatially resolved below the diffraction limit of light. Spatial trajectory filtering is applied to quantify and classify dynamics as either CTRW, the cyan color one, or single-site adsorption-desorption, the magenta color. In short, if a transferrin molecule moves more than 22 nanometer from the initial localization position during a trajectory, a length scale greater than the localization precision, the trajectory is classified as CTRW. Tracking results that increased salt concentrations resulted in a relative decrease of CTRW of transferrin at nylon. Although the transition to pure adsorption-desorption is not complete, the observed salt-dependent changes in surface dynamics hold important implications for the ensemble immersions and simulations. Single-frame displacement distributions quantify the shift toward immobile adsorption events at high salt concentrations. Analysis of the single-frame displacement indicate that a decrease in frame-to-frame hopping is observed as salt concentrations are increased from 0 mm to 1000 mm in a tunable patient. Single-frame displacement analyzed indicate that after the initial adsorption of a transferrin molecule, the likelihood of transferrin exploring nearby sites is decreased at high salt concentrations. Next is single-molecule transferrin kinetics. Transferrin adsorption rates increase on the nylon interface at higher salt concentrations, reaching roughly a 10-fold increase at a salt concentration of 500 mm. The rate of adsorption is quantified by counting the number of new identified molecules that arrive at the interface per unit area. Increase in adsorption rates and increased ionic strengths correlate to more transferrin molecules imaged at the surface. Quantifying the absolute adsorption rates is often experimentally unachievable, but can be easily achieved with single molecule tracking. The increased binding at higher salt concentration can be explained by decreased solubility of transferrin molecules in the mobile phase, as predicted by Hofmeister. Increased adsorption rates indicate that the transferrin molecules exhibit a greater attraction for the nylon interface as salt is increased in the mobile phase. In addition to adsorption kinetics, single molecule results also elucidate desorption kinetics of transferrin. Surface residence time analyzed uncovered two distinct populations of transferring desorption from the nylon interface, revealing an overall increase in desorption rates accompanied by a decrease in rare long-life events. Surface residence time distributions are represented as cumulative distributions functions to uncover rare long-life binding events. Visual inspection of the tails of CDF distributions highlights that increasing salt concentration decreased the number of rare long-lived adsorption events at nylon. The dominant desorption pathway of transferring is changed at salt concentrations greater than 10 mm. Desorption is dominated by fast dynamics at salt concentrations greater than 10 mm in comparison to slow kinetics in the lower and no salt conditions. The fast desorption population is dominated by molecules undergoing single-site adsorption-desorption, given they desorb quicker from nylon in comparison to transferring molecules undergoing a CTRW. The slow desorption component in the kinetic analysis most likely represents the CTRW fraction, which is supported by the strong correlation between the decrease in A2 and the decrease in CTRW. CDF fit results show the desorption rate constant for the fast population increase as a function of salt, whereas the slow population desorption rate constant is unchanged, leading to an overall increase in desorption. These results highlight the utility of single-molecule techniques to quantify kinetic metrics in highly dynamic and complex chromatographic systems. Last for my part is Monte Carlo chromatographic simulations. These simulations are underpinned by the stochastic theory of Giddings and Airy, which reveal that the kinetic data extracted from surface residence time fits predicts better resolved chromatographic peaks as salt increases. Simulated chromatograms are generated using the pre-exponential coefficients and desorption rate constants found from the experimental fits of the surface residence time. Increases in salt show a qualitative thinning of the profile and a visual decrease in the formation of a chromatographic tail. Measurements of the FWHM of corresponding peaks support the mobile phase increase chromatographic efficiency via profile thinning. Symmetric broadening or thinning is a direct result of the number of interactions that a protein has with the stationary phase surface. The combination of the simulations and single-molecule results indicate that mobile phase effects are major contributors to peak symmetry. The simulations illustrate broadening produced by the measured kinetic difference isolated from mobile phase effects. Experimental FPLC measurements provide a convolution of both kinetic and mobile phase contributions to peak shape that cannot be separated empirically. By peak aligning, the Lucian contributions are removed from the mobile phase time and bright retention times to emphasize the broadening effects predicted by desorption kinetics. Exclusion of other effects from the mobile phase guarantees that changes in the chromatographic shape arise from kinetic differences alone. Thank you, that's all from me.
Greetings, I am Joyce here and I shall carry on with the advantages and disadvantages of salting out in the protein polymer membrane interaction in conjunction with the mechanistic examination. Well, so as to begin with, for the advantages of the salting out that is done in protein polymer membrane interaction is that it can elute the desired component of the protein mixture by quantifying the interaction and kinetics of single proteins at the surface of the membrane as the salt concentration is increased. Next is its increasing the salt concentration has led to an increase in adsorption and desorption of the kinetics. This can be observed at the nylon stationary phase. The electrostatic shielding likely dominates the adsorption rate as it increases, while the increased overall rate of desorption that includes structural changes at higher salt concentration. And lastly, from salting out, the altered desorption kinetics were not observed with fatty acid molecules, which highlights the complexity of large biomolecules that exhibit complicated dynamics, such as unfolding. This simply means that it minimizes the unfolding stress to the protein encapsulated. As for the cons with other disadvantages, in addition to the precipitating of the specific protein of interest, the contaminants are likely to get precipitated as well. So Due to that, to obtain a purer protein of interest in this study, additional purification methods such as the ion exchange chromatography was done. Alright, proceeding to the conclusion. Like all steps in the purification process, the salt concentration is adjusted empirically in the mobile phase to elute a desired component of a protein mixture. So actually, by quantifying the interactions and kinetics of single proteins at the surface of a membrane, as the salt concentration is increased, we relate the mechanistic nanoscale observables to an improvement in the peak broadness observed in the real separation. So this means there is sufficient quantitative description obtained when the salting up process is allowed for predictive optimization. Single molecule surface transport modes and kinetic analysis coupled with ensembles have guided a mechanistic explanation for the increased separation efficiency observed in the bulk separation that occurred during the salting up process. The simulations informed by the small-scale single-molecule observation is to be used to optimize separation conditions, leading to more efficient separations. Thus, predicting peak broadening and single-molecule kinetics is further supported by simulation of elution profiles. Then, with the increasing prevalence of single-site absorption-desorption of the transferrin at nylon is directly modulated with salt concentration. So increasing salt concentration led to an increase in adsorption and desorption kinetics at the nylon stationary phase. So the electrostatic shielding, as mentioned before, dominates the adsorption rate, while the increased waiting times are attributed to the surface-induced structural changes to transferrin at higher salt concentrations. The reduction in chromatographic broadening found in the ensemble separations of transferrin is explained by a decrease in the rare long-lived binding that correlates to a decrease in the continuous time random walk CTRW motion. Well, not only that, the findings also emphasizes on the importance of understanding the physiochemical and changes of the salting up processes that can have on the protein behavior in benchtop and industrial scale separation. The designs of stationary phase supports the surface anomalous continuous time random walk CPRW diffusion at the stationary phase, which will lead to the improved separation efficiency. Well, next, this would be the reference for the research journal that we have looked into for this presentation, entitled A Mechanistic Examination of Salting Out in Protein Polymer Membrane Interactions. And this would be the division on members' contribution for each of us in our group. So whereby for SALIHAH, it was the opening of presentation and members' introduction on the research journal of the chosen downstream process. Then proceeding to Zharif, he was in charge of explaining on the background of research, describing the objective of the research as well as video compilation. As for AMIRAH, she provided the research journal for the presentation and did the explanation on the results and discussion of the research we have done. Lastly, myself, JOYCE, I took charge on the part of advantages and disadvantages of the chosen downstream process, conclusion of the presentation, and as well as this slide, which is the members' contribution. Well, that's all from us. Thank you very much for watching.